in-person and online attendees. Welcome to the first seminar of 2026 for the Australasian Fluid Mechanics Society seminar series. And for the first session today, we have Professor Timm Krüger from the University of Edinburgh. And what is yours, Tim?
Yeah, thank you so much, Morgan, for the introduction, and thank you for inviting me here. Some of you know that I also attended a conference earlier this week in Sydney, so I'm not just here in person from the UK for today's seminar, but of course I'm very happy to give another talk about a different topic actually. And today I would like to show you something that is previous work that we have done over the last few years. And it is combining fluid mechanics, simulation, also experiments. I will not say that much about the experiments, and biology, because it's about blood flow on the microscale, where we need to consider the red blood cells flowing in capillaries, for example. And depending on how much time I have left at the end, I prepared 3 different example cases that I can talk about, but I might just pick 2 of them. So let's see how that goes. But the theme is really that I would like to convey to you the message that a big question in the study of blood flow is a better understanding of the relationship between the structure of blood vessels and the function of tissue or the organ in which the blood vessels are located. Okay, so before I give you an overview of the actual presentation, just a bit of information about the research group. We have been doing research in several areas, all related to fluid mechanics. The first one on the left is microfluidics, which is mostly focused on blood flow, but it can also be particle manipulation, typically in artificial geometries like microfluidic devices. And in our group, everything we do is based on simulations, computer simulations. Whenever you see experimental pictures later on, this has been done by collaborators. So I really like to collaborate with people who are able to do great experiments, because I think it's very important that we combine our experimental and computational expertise to get the most out of a given research problem. In the middle, blood flow. So this is about blood flow in realistic geometries, unlike microfluidics on the left-hand side, which is more focused on in vivo geometries, in vitro, where you have artificial geometries. And I will focus today much more on the column in the middle, blood flow. Also, what we do occasionally is something that you could call complex flows, where you have fluid mechanics, but you also have additional physics, for example, surface tension effects and so on. Just 2 examples. At the top, you see a deformable capsule at a fluid-fluid or liquid-liquid interface. And then the interplay of elasticity of the capsule and the surface tension between the 2 liquids, together with a contact angle at the surface of the capsule, can lead to interesting physics. And at the bottom, you see something that I have contributed to quite a few years back, maybe 10 years now, where we looked at ternary liquid mixtures that are immiscible in this case. So you have mutual surface tensions, and depending on what the density is or the concentration of the phases and the surface tension ratios, you get funny phase-separated shapes that could mimic, for example, what happens in biological cells. But I will not really talk about that today. And while I'm here, I'd also like to acknowledge funding from the Engineering and Physical Sciences Research Council in the UK in particular, and the European Research Council. So the structure of my presentation, I will first motivate why it's important what I'm going to tell you about today. Then I dig in a bit more detail, telling you about blood flow on the microscale. What I will not do in this presentation, I will not give you a very technical overview of the methods they're using, because this is not really a simulation methods talk. It's more a simulation results and application talk, if that makes sense. I mentioned that I prepared 3 examples, all taken from specific papers that we published a few years back. And depending on how it goes, I will talk about 2, maybe all of them. And then, of course, conclusions. So, introduction. Blood flow in health and disease. Now, what you see on the left is the blood vessel structure in a human placenta. And the human placenta is a very interesting organ because it's transient in nature, of course. It develops over 9 months. And it's also extremely difficult to study because typically when you have physical access to the placenta, that is after the delivery, but then because of the delivery, also the structure can change. And therefore, it's very difficult to actually say how the placenta looks like under normal conditions. And what you cannot do, you cannot just image during pregnancy the placenta with CT scans, for example. I mean, nobody would do that, right, with a baby in the womb. So it's extremely difficult to get structural information about the placenta in health and disease. But of course, we can look at that, for example, after delivery. Placenta is, by the way, one organ where animal models are not very useful because in other mammals, the structure is quite different from the human placenta. So you don't learn that much about the human placenta when you look at animals. So here what you see is a healthy placenta. And you can imagine that the structure of the blood vessels will determine the function of the organ. And this is only one example. You could also think about the liver or the brain or whatever. And the reason for that is that you have this network of blood vessels starting at the arterial level, where blood is transported very quickly. So it's basically a highway to transport blood cells to certain areas of the body quickly. But there's no transport of oxygen to the tissue or glucose to the tissue. It's a very, very small blood to tissue transfer in larger blood vessels. So all this transfer that is basically diffusion dominated happens in the region of smaller capillaries. So you want to transport the red blood cells and whatever else you have in blood quickly to the capillaries, then you have a slightly higher residence time and you have much closer contact of the blood cells with a blood vessel wall, where then diffusion can actually transport everything out of the blood vessel. And of course, collect it back because you want to collect waste products as well, like CO2 and other chemicals. And if that structure doesn't look right, whatever that means, you can imagine that the function of the tissue or organ will simply not be proper. But it's extremely difficult to say, well, for a given structure, what does it mean for the function? It's a bit like an inverse problem. If you, for example, observe there is some kind of disease, can you infer what the structure would be for that disease? Or if you see the structure, can you guess whether there is a condition that should be treated or not? So one of the biggest questions we have is the prediction. How do we predict the function of tissue or organ based on the microstructure? And that becomes even more interesting when you look at disease, because so far I just told you about healthy placenta. And well, many people would say they are not interested in blood flow as long as it flows well, right? But if something is wrong, then of course there's a much higher level of interest, typically, scientific interest. And cancer is, of course, a very big player in disease. So for cancer, you can imagine if you have a solid tumor growing, it requires volume and space to grow physically. And this means it can compress blood vessels, for example, in the vicinity. That will change the way blood is distributed, but also the structure of blood vessels inside the tumors is different simply because tumors, when they grow, they release certain chemicals, for example, VEGF, which is a vascular growth factor that can then recruit new blood vessels into the tumor, because tumors typically have a very high glucose and oxygen demand. And in disease like cancer and other diseases, we would like to know what can we learn, not only in terms of fundamental science, but also in terms of treatment options about the relationship between the structure of blood vessels and the function of the organ or the cancer in this case. So the general question is clear. We do not just want to understand how a blood cell moves in a vessel. We would like to understand how other micro and macro scales linked in terms of biological function. This is only an example for the placenta, how you could go from the microscale to the organ scale, taken from a review that we have written a few years back. So for example, at the microscale, you might look using CT at the structure of the blood vessels, and you might develop microscopic models for how blood flows in small parts of tissue or organ. You might scale that up so that you have a model that is working at higher or larger scales, but maybe less detailed. That can be validated using MRI, Doppler imaging, and so on, which has a much lower resolution, of course, than micro-CT. And in the end, you could then ask, okay, what can we learn about the entire organ? And potentially even, how can we design an artificial placenta, right? Because if we understand how it works, in principle, we could also design one to support cases where naturally it would be very difficult to deliver the baby healthily, right? So this is only giving you an idea that you have different scales and you can imagine there's a flow of information and so on. This is not very straightforward. So a bit about microscopic blood flow. If you imagine a blood vessel, then of course blood coming from the heart is first in a large blood vessel, and then you have bifurcations, and you have more bifurcations and so on. You might have more complex geometries than that, but in a simple picture, you have a larger vessel, in this case coming from the left, or the blood flow is coming from the left. You have a bifurcation point, and each blood vessel will then bifurcate again until you reach the capillaries. So this would be the arterial side, and after the capillaries, you are collecting the blood again into the veins, going back to the lung and the heart. Now, interestingly, if you look at blood vessels which are a bit smaller, like this one, let's say around 50 micrometers diameter or so pretty thin already. Each red blood cell that you can see here has a diameter of around 8 micrometers, okay? So you can see already there is some heterogeneity in the distribution of blood, and that is simply because we have a confinement effect here. The size of the blood cells compared to the diameter of the vessel is not negligible. And in particular, in these parts of the network where you're a bit closer to the capillaries, these size-based effects play an important role because, interestingly, what happens now, if you measure the total flow rate of blood going into the two branches of a bifurcation, so let's say here and here, of course, the total flow rate has to match the flow rate in the feeding branch. But if you work out the split ratio, and now only look at the red blood cells and work out their split ratio, they're not the same necessarily. And that is because at that scale, blood is not a homogeneous suspension. You have this kind of layering effect where you get the cell-free layer close to the surface of the vessel. And the cell-free layer, where the question is it really free of cells? But it's a cell-depleted area, basically. And that is because red blood cells are soft and they tend to experience lift forces, pushing them away from the wall, which has been studied extensively in experiments and simulations. But due to shear-induced diffusion effects and, of course, collision effects between the red blood cells, they try to disperse again, and there will be something like a dynamic equilibrium, more or less, leading to a non-trivial distribution of the density of red blood cells, number density, across the cross-section of a blood vessel. And typically you see a peak in the center. You might have additional layering peaks in between, but you normally drop to a very low concentration in a region a few micrometers thick next to the wall. Now, this region will, of course, affect what happens at the next bifurcation, because if the cell-free layer is of a comparable size to the thickness of the blood vessels, not equal size, just, it's getting relevant, then you can already see that there is some kind of bias typically affecting the smaller blood vessels, where you get a smaller fraction of the red blood cells compared to the total flow rate of blood. And what I mean by total flow rate of blood, it's blood cells and blood plasma together. Blood cells, 45% by volume, plasma, 55% by volume. So you see, or you can guess by looking at the first bifurcation, there's some kind of effect driven by the heterogeneity of the suspension, leading to a specific split of the blood cells in bifurcations. Now, sorry, wrong computer. This has been studied in great detail by Priest and Secomb in already the 90s, and they developed a phase separation model. So the two phases would be the red blood cells and the plasma. And I don't want to go too much into detail here. It can be quite technical, but those of you who are interested, you can of course read about that. Based on the diameters of the blood vessels, the feeding diameter, here shown in red, and then you have the branches alpha and beta in green and blue that go to the right. All of them might have different diameters. All of them might have different flow rates for the whole blood. Based on that, it is possible to predict what the fraction is of red blood cells going from the feeding branch into the child branches alpha and beta. And you would naively expect, if you plot the red blood cell fraction going into each branch versus the total suspension fraction, you should have a straight line going from 0, 0 to 1, 1, because this you would have if you had a perfectly homogeneous suspension at every scale. But because of this finite size effect, you can see that you get a biased curve instead that tells you at relatively small fractions for a blood flow, you might actually have no red blood cell going into a branch, right? So if you look at, let's say, the area 0.1 on the x-axis, you still have 0 for the red blood cells. And this means you get a plasma channel where you just have blood plasma going into one capillary, for example, okay? And in total, you have, of course, mass conservation, which means if you take the flow rates of branches alpha and beta, put them together for the plasma and red blood cells separately, mass is, of course, conserved. But these curves, they have been well validated. We actually ran a few resolved simulations also with our codes, and we confirmed that we get essentially the same results in our simulations. So these are well-established results under certain conditions, because you need to make some assumptions to get there. And my first example is actually about what happens if these assumptions are not correct anymore. Yeah, so the main takeaway message here is you don't have to understand at this point why the curve looks like this. You can read about that in the original papers. The main message is you don't have the diagonal line. You have a function that looks different than a straight line. And that means you get different volume fractions of blood in different branches. And that is simply a size-based effect. Size of the red blood cells compared to the vessel. So, blood flow modeling. I just told you something about blood flow and why it's important. But now let's ask, what do we need to do if we want to model blood? And by modeling, I mean towards setting up simulations and running computer simulations. Well, how do you run, or how do you set up a computer simulation of blood flow? The first question you should answer, of course, is what is the scale of relevance for you? What are you interested in? Because you have, in the human body, you have blood flow. Let's start at the top right. In the largest vessels, where you might just get away with a single phase non-Newtonian model. Some people say you need a non-Newtonian model. Some people say you can also have a Newtonian model. It gives you relatively good results, but it would be a homogeneous blood flow model where you might have shear thinning, right? But you would not consider, other than the rheology itself, the effect of the red blood cells. So the red blood cells will affect the viscosity of blood, but you would not care about cell-free layer and distribution of blood cells in the blood itself. And that is typically good enough if you look at arteries, because the diameter is much, much larger than the diameter of a red blood cell. If you go to the bottom left, you are approaching smaller scales, both in terms of vessel diameter, which is shown on the x-axis, and typical flow rates and also Reynolds numbers, which you have on the y-axis. And what I'm telling you about today is more in the bottom left corner, where you have the, let's say, the two pictures here of blood cells in a small vessel and blood cells entering a constriction. So this is really the area of interest for this presentation. And for that, you cannot simply assume that blood is homogeneous. I told you that already. You cannot disregard the red blood cells as objects in flow. And you somehow need to resolve, to whatever that means, resolve or capture individual cells moving in a Newtonian fluid, which is basically describing the plasma. So you have blood plasma plus particles inside, which you need to simulate. Okay. So I think I have one slide showing the methods we're using for that, but I will not give you technical details because I think it's not the right presentation for that. So what we do, we have a fluid solver for the blood plasma and also for the fluid inside the red blood cell, because red blood cells are essentially capsules filled with a hemoglobin solution, which again is more or less a Newtonian liquid. We use the lattice-Boltzmann method for the flow, for the fluid, which lives on an Eulerian lattice, which is visualized here by the cube, by the gray cube, right? So you have a lattice which is fixed in space, and you solve for the fluid flow at each grid point, right? But these points are not moving. They are just fixed in space. For the red blood cells, we use a membrane solver, which is a finite element approach, which is Lagrangian, which means that the red blood cells move, of course, and they have meshes. So each red blood cell we have has a mesh, and these meshes deform and move along, and we track how the meshes move. So this is why it's a Lagrangian approach. And of course, we need to couple between the two, and it's a two-way coupling, which means the flow is affected by the red blood cells and vice versa. For this, we use the inverse boundary method. And for all the simulations you see today, we have used the HemoLB blood flow solver, which has implemented all these methods. Okay. And then when you run these simulations, you might get something like this. I will show you a few videos later, where at the top you see red blood cells in a network of capillaries in a retina. And at the bottom, you see something that we haven't published yet. That's simulation of blood flow in a reconstructed geometry of a placenta, a subset of the placenta. So that's the background, and I see that we have already spent 25 minutes or so, and therefore I'm not entirely sure I can show you everything here. But let's start with maybe the distribution of red blood cells in consecutive bifurcations. And you remember I told you that you have this particular flow split between different branches and so on. So this is based on a, or not based, it's taken from this manuscript to this paper that we published already 6 years ago. You can see there are quite a few people involved here. And of course, I want to thank all of them for putting together this great manuscript. My contribution was more on informing and discussing the microfluidic simulations, not the experiments and biological implications. But if you're interested in this particular work, so please check it out. It's definitely worth a read. And here, the key question is, what role does the arrangement of blood vessels play in tumors where you have certain abnormalities, which maybe I don't want to go into detail too much at this point to save some time. So let's just say that in healthy vasculature, you typically have a distance between consecutive bifurcations, which is sufficiently long for the cell-free layer to recover after each bifurcation. So you can imagine if you have a bifurcation, then the distribution of blood cells on the cross-section will be distorted, but due to collisions and lift forces, and you will have a redistribution of the blood cells. So after some length into the child branch, you can expect that you don't know anymore that there was a bifurcation upstream. Okay, and at some point you will get the next bifurcation. But typically in the vasculature, at least well on the arterial side, you have quite often bifurcations far enough away from each other that you don't really see the impact of the previous one. And we know that in certain tumors, that is not the case, where bifurcations can be much, much closer to each other. So keep that in mind. The flow-wise separation distance between consecutive bifurcations. So what we did, and this is really simple in terms of simulations, it's really, really simple. We looked at 3 different cases. The first one on the left, we have 2 consecutive bifurcations. These are straight tubes, basically. I wouldn't even say these are blood vessels, right? These are just tubes. They have a diameter of 33 micrometers, internal diameter. So you can only see the outside, basically, but there's blood inside. And branch 0 is the overall feeding branch. You have 1 bifurcation giving you branches 1 and 2. And this bifurcation is set up in a way that all the diameters are the same, but also all the flow rates are the same, which means branches 1 and 2 have the same flow rates, which are half the flow rate of branch 0. And then branches 3 and 4 have the same flow rates, which are half of the flow rate in branch 1. So a quarter of branch 0. Okay, very simple flow split, same diameters. And the pre-second model would predict that you get also an equal red blood cell split because you don't have any asymmetry in terms of diameters or flow rate. So from a physics point of view, you should have exactly the same volume fraction of blood in all branches. Okay, so this is what we looked at. And we measured the hematocrit, so the volume fraction of, or it's basically the flow rate of red blood cells by volume over the total flow rate. So it's called the discharge hematocrit. It's really the flow rate ratio. It's not the volume fraction measured by looking how much volume is covered at a given time. And you can see that to quite high accuracy, of course, we have a finite number of red blood cells flowing in the simulation. You can see that the hematocrit in all 4 branches is basically the same. And it is very close to 20%, which is our inflow hematocrit in branch 0. So that is a confirmation of a very special case where we know what the answer should be. Great, that works. Now the question is, if you move the bifurcations closer together, and in the first case, it was 25 times the diameter, roughly. In the second case, it's only 4 times the diameter of the vessel. So question to the audience, and maybe for the online participants, because I can see the chat also, if you want to guess which branch in the second case, so in the middle column, which branch could have a higher or lower concentration of blood cells or flow rate of red blood cells? Any guesses?
4 higher.
Itai, which one?
I said 4 higher.
You say 4 is higher.
That's my intuition. In the middle one. I don't know why, but that's my feeling.
I can tell you that branches 1 and 2 should be the same still because the condition branch 0. So I can tell you branches 1 and 2 will be identical, right? The question is really what happens to branches 3 and 4. So you say branch 4 will have a higher hematocrit. So we don't have somebody else says 3 higher. So okay, that's getting interesting. Okay. Okay, and then let's move on to the I'll show you the results a bit later. Let's look at the case on the right-hand side where we have a flipped geometry. So the second bifurcation goes the other way, but everything else is exactly the same in terms of boundary conditions. So any guess here what happens in this case? Opposite.
3.
Okay, so let me show you the results.
That was good.
For the second case, you can see that branch 4, the one that goes up in the second bifurcation, has a significantly higher hematocrit, and branch 3, the one that is going straight through, has a lower one. And that is certainly caused by the small distance between the two bifurcations. And interestingly, when you now flip branch 4 over to the bottom, then you see that branch 4 gets fewer red blood cells. So it's actually the branch that stays at the top, basically. So compared to branch 4 on the right-hand side, branch 3 is at the top, right? Because 4 goes down. But in the middle, branch 4 goes up and 3 is straight. So the one that tends to go up has a higher hematocrit. And if you think about it, that is actually not a big surprise, because this is now how the red blood cells look inside. At the first bifurcation, we have an even split, but you can see that in the segment between the bifurcations, there's basically no cellular layer anymore at the top boundary, and there's a much higher one at the bottom boundary. And this affects, of course, how the red blood cells flow into the next branch at the second bifurcation. So there is a bias for red blood cells to pick the higher branch, the one going up, because there is no cell-free layer on that side. Okay? And the pre-second model assumes that at any bifurcation, you have a fully developed cell-free layer. So when your cell-free layer is distorted by whatever happened before, or let's say more generally, if the red blood cell distribution on the cross-section is distorted just before the bifurcation, then you cannot expect the pre-second model to give you the correct result. And there are a various number of situations where this can happen, and this is a very simple one. I mean, this is ridiculously simple what we have done, right? So essentially, that's
Tim.
Yes.
Think about the streamlines of the plasma as a driver of shear on the red blood cells that may explain that. The kind of how I was running in my head.
So the thing is really that if you follow the streamlines, you have more red blood cells in the region where the streamlines go into the branch going up. So it's basically an advection thing, right? And that is
no shear through the.
You have shear, but that is not really relevant here. It's you can essentially look at the number of red blood cells in certain regions of the flow, and look at segments of the flow going in one branch and into the other. Now, streamlines are a bit difficult here because we have a suspension.
Sure.
You can define the streamlines on average, right? But of course, you can look at that, but it's really mostly about the spatial distribution of the red blood cells. There's not that much dynamics happening. Also, if you change the softness of the red blood cells, for example, for the same distribution of the cells, you will get a very similar split. So it's not the dynamics of the red blood cells in the bifurcation that is extremely important. I will show you a different example where this is different, but in this particular case, it's, you will be mostly determined by where are the red blood cells at the point of the bifurcation. Are they more biased to one side or the other side? Okay, so this is only a message I want to send to you that reduced order models, like the pre-second model, very powerful, but also limited in their applicability. You really need to know whether the conditions and assumptions originally made are still valid to use a specific model. And yeah, so the question could be, how can we augment the pre-second model under certain conditions, for example? What I want to show you now is some work we have also done together with experimentalists, and this is about lingering of red blood cells in realistic bifurcation. So I will tell you what lingering is. This is based on a paper that we published 3 years back in the Biophysical Journal. Also a very nice collaboration with Christian Wagner's group in Germany, who did the experiments. And the experiments were in place before, and then we had discussions about the results, and we said, oh, we can actually run accompanying simulations. And we found that based on these simulations, we got a complementary view of what is happening, and it led to this very nice paper. So what is lingering? Here, first of all, you see at the top 3 different bifurcations in vivo. So this is where blood cells are tracked optically in vivo in a hamster. And at the bottom, you see the same geometries reconstructed in the HemLB code with red blood cells flowing in the same direction. And the question is now also how the red blood cells distribute in this bifurcation or these bifurcations. But one particular question is what happens when these red blood cells, they do not directly or immediately go into one of the branches. They first huddle a bit with the apex of the bifurcation, which is called lingering. Because the red blood cells linger around, they stay for some time before they go into one of the 2 branches. And you can now, of course, think that depending on how the apex of the bifurcation is interacting with the red blood cell, and you can see there is a deformation effect here, the red blood cell might have a higher tendency to go into one branch or the other branch, independently of what the pre-second model says. And in fact, we found that the lingering intensity, I will get back to that, what it is, will tell us to what extent we deviate from the pre-second model. So pre-second, they did not consider lingering in the model, but in reality, we have lingering. And maybe I can just show you a simulation, why it's actually 2 simulations. So these are 2 separate simulations in the same geometry. The only difference is, I think it might be, well, the concentration, but at the top, the red blood cells are softer, and the blue red blood cells at the bottom, they are more rigid. And if you now look at the red blood cells approaching the apex of the bifurcation, you can see that some of them stay a bit longer, and it takes a bit of time for them to go into one of the branches. And depending on how many cells you have, because you have the next one pushing from upstream, and how strongly they deform, you will, of course, have different results, different outcomes, okay? And you could say, well, on average, it doesn't matter, you will have the same as before. It's not exactly true. On average, you will not have the same. Okay, so this is how it looks like. And now, how do we define lingering? Because if you don't know what lingering is mathematically or physically, then it's difficult to work with it. And well, saying what lingering is basically saying what the length of a piece of string is. It's, well, how long do you need to wait until you say this red blood cell is lingering? And how slow does it have to get in order to linger? There's no clear definition. So what we have done, we looked at the velocity probability distribution of the red blood cells approaching the bifurcation. And maybe it's a bit easier to look at the simulation results. At the bottom right, you see a relatively shallow gray line, which is bimodal. So I have 2 maxima. You see something similar for the experimental results at the top, but you have a much stronger bias towards higher speeds, but you still have a somewhat bimodal distribution. So what we just said, well, bimodal means in this case, you have a bunch of red blood cells which are fast going through the bifurcation, and we have a bunch of red blood cells which are quite slow. And if we now say we have these 2 groups, if we just take the minimum between the 2 maxima and say everything below that speed is a lingering red blood cell. This is how we define it. You can challenge that assumption. Of course, we can look at the results if we define it differently, but this is what gives relatively good results for the discussion. So essentially, the vertical dashed line you see in both plots defines the threshold between lingering cells on the left that are slow and non-lingering cells which are fast, going quickly through the bifurcation. And on top of that, which I didn't say, on the left, you see these red circles near the bifurcation. So a lingering red blood cell also has to be within that circle. Otherwise, it would not be considered as a lingering cell, because for whatever reason, you might have a slower cell in a different part of the geometry, but those are not counted as lingering cells at the apex. Okay, so now what we did, and I really don't want to go too much into detail, but you see a lot of information here, but I just want to distill this to the essential. And if you're interested, please look at the paper. We defined a lingering Peckley number. And Peckley number is basically a ratio of timescales for different transport modes. And you can say, okay, let's take the average lingering time tau and divide it by the average advection time of the red blood cells in the bifurcation area, which is the average speed of the red blood cells divided by a typical size, like the radius of the vessel or radius of a red blood cell. So it doesn't matter that much as long as it is a fixed value. And the Peckley number is simply the ratio of these two timescales. And this means if there's no lingering, tau is 0, the Peckley number is 0. And if there's a lot of lingering, the Peckley number is high. Okay, so whenever you see the Peckley number on the x-axis going to the right means you have more lingering. On the left, you have no or weak lingering. So essentially, what we did, we looked at the distributions that were predicted by pre-secom, which you see as solid lines at the top left, for example. And you see the square that is for the, well, MD means main daughter. It should probably be the main child. And SD would be the secondary child. So you have one branch that is typically favored in terms of flow rate, that has a higher flow rate, that is the main one. And the secondary one has a lower flow rate. So you can see the pre-second model predicts these solid lines. And the data points with arrow bars, these are the experimental measurements at the top. We also have, so this is experimental. We also have simulations for that. And if you now look at how is that flow split different from the predicted pre-second split, this is what we call lowercase delta ZF, Zweifachfung effect, which is basically the effect that we have been talking about before. So if you look at the bottom left, the y-axis, delta ZF, tells you to what extent you deviate from the phase separation model. And the x-axis tells you how significant lingering is. And as you would expect, zero lingering should lead to a zero deviation from the model, and that is what we have. And the more lingering is important, the more you deviate from the predictive model that I introduced to you at the beginning of my seminar talk. And at the bottom left, this is experiments. Bottom right is simulation. You can see a very similar trend, but a different range of parameters. For example, in simulations, we did not have as much lingering compared to the experiments. And we believe that is because we did not have a very good model for the contact between the red blood cell and the apex. So in reality, you have a glycocalyx, you have these polymers, and there's all kinds of friction, physical friction between the red blood cell and the endothelium, which is normally not important for blood flow modeling because red blood cells typically keep their distance from the endothelium, self-layer, right? But at the apex where they collide, it's different. And we don't have good models for that, for simulations. So we believe that the difference, the quantitative difference between experiments and simulations is due to that. But you can see there's a qualitative similarity between experiments and simulations, which is very encouraging. So we are able to capture the main driver between the lingering activity and the deviation from the pre-second model that I...
So what are you changing in the input? Is it the softness of the red blood cells? What
it could be the softness, it could be the different bifurcations, because you can see here at the bottom.
So it's many different conditions.
There are, I think we have 12 different bifurcations that we looked at. And you can change certain parameters, and based on that, you get different data points. And for each data point, if you measure for some time, you look at your red blood cells, you get your distributions, you define your lingering criterion, which determines what the Peckley number is. And you look at the distribution of the cells and compare that with the expected distribution if there was no lingering. And that gives you the Y component of the data. Right? And the interesting thing is that you have a clear impact here. Right, and the question is, why is lingering changing the distribution of the cells? So, or where is the lingering coming from, actually? And we looked at a number of different metrics. So if you have all these geometries, which are taken from in vivo microscopy, of course, we didn't design these bifurcations. We just got these bifurcations, okay? And now you can start thinking about how are they similar, how are they different? So what are the key features of these bifurcations? And we looked at a range of parameters and found that two of them are particularly significant when it comes to affecting the Peckley number. So one of them is the curvature of the tip or the apex of the bifurcation. If you have a higher curvature, so you have a more pointy apex, you tend to have much more lingering, which is somehow obvious because if a red blood cell is nearly pierced by the apex, it is much more difficult for the cell to roll over to one side. But if you have a relatively flat apex, the red blood cell can quite easily slide to one side. So that is what we believe is one mechanism. Well, there is a strong correlation. It is not very clear-cut on an individual basis, as you can see. But we definitely believe, based on these data, that the apex shape, so really the shape of the apex itself, is important. The other one is the ratio that doesn't really tell us much about the mechanism, but it's encouraging what we see here. It's the ratio of the flow rates in the two branches. So if you have a very similar flow rate going into two branches, then red blood cells that are initially in the middle of the feeding branch have a much higher chance of hitting the apex because the flow is split in an equal fashion. So red blood cells will just go to the apex because they don't know until the end where to go. And then they have a much higher probability of contributing to lingering. So you can see if the ratio is about 1, which is equal flow split, we have the highest lingering probability. But if we have one branch that gets most of the flow, the red blood cells are early on dragged into that branch, and they have a much smaller probability of reaching the apex, okay? So the lingering effect is much smaller. And this means we can, if we look at the geometry alone, and we know what the flow rates are, we can to some degree already predict how important lingering will be, and to what extent the pre-second model will be valid or not. We did not really develop an extended model for that, but it was basically a door opener to see to what extent is it important. Okay, so I think I might actually just very quickly show you what else we have done, but I don't have time to go too much into detail. This is another work also with experimental collaborators, and we are particularly proud on this one because based on that, we were able to give very clear statements about what the relationship is between the blood flow in a capillary network and the development of that network over time. Because normally you would expect if you simulate blood flow, well, it's a fixed geometry and it never changes. But in reality, capillary networks can and do change over time, in particular if it's a developing network, which is, for example, in this case, we had a mouse model and we looked at the mouse directly after birth or a few days after birth, where you have a lot of redevelopment of the network. And the question is, if you imagine you have a capillary network or vascular network, and it changes over time for whatever reason, what extent is the blood flow inside the network responsible for the redevelopment? Is the redevelopment just imposed by some, I don't know, given process, or is the blood flow inside the network also contributing to the redevelopment? Okay, and we were able to answer that question to some extent. So I don't want to go too much into detail, but we looked at the capillary network in the retina of a mouse, and we were able, because the eye of the mouse has to be sacrificed for that to have the high resolution scan, which means you cannot measure the same network repeatedly over time. But if you look at different molecular markers, in particular, if you look at endothelial markers and you look at collagen markers, you can say something about how the network looked like a few days before. So we are able to draw the same network twice at different points in time. And that determines how it has changed in the meantime. Okay. Now, the question is, how is it related to the blood flow inside? And here I just want to show you a video. So that's one region of interest of the larger network, where we cannot simulate red blood cells in the entire network. So you can see that there are quite a few branches here that are devoid of red blood cells. Others are nearly like highways. We also have one red blood cell that is stuck at the top. Yeah, and you might guess that those branches that don't have many red blood cells inside are the most useless branches, because they don't contribute to the transport of oxygen. Because oxygen is transported inside the red blood cells before it's released to the tissue. So in terms of oxygen transport, any capillary that doesn't, over an extended period of time, have red blood cells inside, it's not doing its job, right? And then what actually happens, and I won't show you the results in detail, but we found that those branches that at an earlier time don't have many red blood cells inside are those that disappear later on due to the redevelopment of the network. And that is because, which you cannot see here, the branches are covered on the inside with endothelial cells. So it's a one cell layer thickness, basically, that is keeping the inside and the outside of the blood vessel separate. And these endothelial cells, they are not just passive tissue, they respond to the blood flow somehow. There's mechanotransduction happening. It's quite difficult to say what it is exactly, but these cells can sense, okay, there is some kind of flow happening, there is some kind of red blood cell passing or not. And under some circumstances, the endothelial cells say, okay, I'm not needed here, so I migrate to a different part in the network. They disappear, they move away. They don't just die, right? They move away. And then the blood vessel eventually closes and is not a blood vessel anymore. And we're talking about a small scale. We're talking about micrometer size blood vessels, not a major artery. Okay? And our simulations helped to show that there is a strong correlation between what happens inside a capillary at a given time and what is the fate of that capillary a few days after, during the development of the network. And I think that is a very nice achievement. Okay, so I think I can skip this one. And that brings me to the conclusions. Now, I believe that the ultimate aim in this research area is that we have better models for blood flow at larger scales so that we can say something about health and disease and tissues and organs. Larger scale models also, or reduced blood flow models, because we simply cannot run resolved simulations in all these networks. And even if you could do it, you wouldn't do it on a routine basis. It's simply not the point. So we would like to develop, let's say, more appropriate reduced order models that take into account all kinds of situations like distance between bifurcations, lingering, and so on. Okay? But in order to get there, we need to have microscale simulations because we need to develop these models. I'm not particularly talking about machine learning. Of course, machine learning is also one thing we can apply. But as scientists, we are very interested in understanding why things happen. So I still have some hope that people are, in a few years' time, are still looking at that from a mechanistic point of view, rather than just a data-driven point of view, because we are curious beings. In any case, so no matter what happens, we need data and we need insight that we can only get if you look at the cell level, both in experiments, of course, and computer simulations, right? So that's maybe the key message I want to send, that if you're interested in blood flow modeling, there's so much that still has to be done that is related to biological function of tissue, From a mechanical point of view, maybe most of that has been done. How do the red blood cells flow in a given vessel? How do they deform? How do they migrate due to lift forces? I think that's probably very well studied. But if you now ask biologically relevant questions, there's so much that can still be done. And I hope that this research field will still go strongly in the next few decades or so. Well, thank you very much. And I'm happy to take questions.
All right. Thank you for the presentation. I think for the question and answer part, we'll do it both in person and online. So maybe I will look after questions in person, and if you have any questions online, Travis, our co-convener, will be moderating the questions online. Would we like to start with the discussion in the room?
I come from geotech granular flow sort of background. So in our case, we mainly deal with hard particles traveling in the fluid, and we care for long range interactions when the suspension is dilute, and short range interactions when it is dense. But in blood flow, given the fact that RBCs are flexible, in this sort of modeling approach, both short range interactions and long range interactions are equally important, or is there some sort of balance between how you treat them?
Well, it's because each red blood cell is surrounded by many other red blood cells. Let's talk about the non-dilute regime, right? Because you can, of course, simulate blood at 1%, 5%, and so on. But if you talk about realistic, let's say 20 to 40, 45%, you are essentially in a, well, not dense suspension, but you're in a denser suspension. And then hydrodynamic interactions between cells. You have, of course, volume effects where you can simply not go to a given place because you have a cell already. This is really determining what's happening in blood flow. I think the short-range interactions are much more important here, simply because you're screening long-range effects. I think that a red blood cell at a given point will be much more affected by the surrounding ones than a blood cell that is farther away. And other than hydrodynamic interactions, except for maybe steric effects where you also have some kind of molecular interactions at some point, and we can talk about bridging, depletion, and so on, which is more important at smaller shear rates, because then the cells have a bit more time. There's less stress shearing them off each other. But these are all short-range interactions. So I would say the short-range behavior is much more important in this case.
Tim, fantastic work. I have a couple of questions, if it's okay. It's just like, one is like, maybe like Shivu, it's by analogy. In my mind, I'm really interested actually in the last example, in what you haven't yet done, I think, which is, you mentioned that the network sort of evolve itself. And right, so by the way, in our world, we have meandering rivers, right? So you have rivers that take gradually shape with the earth, and eventually they leave some rivers dead and they just disappear. And that's just like, I'm very, yeah. So do you have any thoughts about how to evolve the network, maybe in a reduced order way, to see how the retina sort of achieve its final goal?
Yeah, so I'm not sure to what extent we can reduce that directly, but what people have done is using agent-based models where each endothelial cell is an entity and there's a criterion or condition for each cell to do something. So if you have, let's say you have a capillary, you have, let's say, 20 endothelial cells inside, then the first one might say, oh, I'd like to remove elsewhere for whatever reason, right? Because certain conditions are met or not met. And maybe there's also some kind of consensus between them, because they might also communicate. And that would have to be established first. And based on that, you could have a reduced order model where you say, okay, I don't look at agents, individual agents. I look at maybe just the length of a vessel. I know number density of endothelial cells, and they have some kind of distribution function, probabilities or whatever. But the problem is really the decision-making of individual cells. It's very difficult to find out why they do something. So for example, what is it actually that they sense when blood is flowing by? Is it the average wall shear stress? Is it the fluctuation of the wall shear stress? So for example, the minimum or the maximum? Is it the difference between the two? Is it the average frequency of the oscillation? It could be anything. So to what extent are they sensitive to high frequencies, low frequencies? Do they sense it over the entire surface or just certain points? If we don't really know that, well, maybe we don't have to know all the details. We just need to have a model that works somehow. But it's a very biological problem. And as a physicist, it's extremely difficult to say, let's make this assumption or that assumption, because we don't have a good justification for the assumptions.
Can't you just like have an evolving network by your experimental colleagues? Then at least you make a decision and you can see how it fits that.
I think what you could do, you could, and this is very challenging experimentally, you could have a network that evolves. You can somehow track individual endothelial cells, but then you wouldn't know what the flow conditions are around these cells, because experiments will not tell you that. You could then have simulations in the same geometry and estimate, okay, oh, there's something interesting happening in terms of wall shear stress. But then you can only correlate observations. You cannot say, oh, yes, it's this one factor or another factor. So I'm wondering whether we will ever be in a position where we can say this endothelial cell is doing something for that reason. And based on that, develop the model. I think probably in the end, it's more phenomenological where we say, okay, under those circumstances, we have a 20% chance of something happening or 70% chance. That's probably more realistic, at least in the short term.
Can I ask another question?
Yeah, sure.
Actually, back to more the aspirational side. So in slide 6, you talked about your review. There was a review about sort of how all the modeling strategies are connected. And what always amazes me, or so the things that in my head is always the difficult part, is transferring from C to B, from B to A. I imagine that those are, and we have typically those things. Is this, are there sort of like upscaling type of, how is this upscaling happening? I can see that vision, but those arrows are not describing how it's done.
Yeah. So. Okay, what I should have said here, this is in an ideal world, how it could look like.
Yeah.
It doesn't mean that all these arrows are materialized.
Okay.
Right? How do you upscale? I mean, you have upscaling approaches. Mathematically, you can define what upscaling is, and you can have a sensitivity analysis and so on. But how to do this in practice? We still don't have everything we need on the micro scale. And this particular project here, and I showed you this one image of flow in the placenta, that was the first step that we took to get a bit more understanding what's happening there, but it's not the end. So this is more like a vision, what you can see here. Partially realized. Right? But
some errors.
You cannot just start here and say, oh, yeah, we go up and in silico placenta, and that's fine. It requires more work.
Do you have a question online, Morgan, if that's all right? One from Zhongzheng. Zhongzheng, do you want to unmute yourself and ask your question, or would you like me to read it out? I can just read it out. He was wondering to what extent the deformation of the vessel itself might impact the flow process.
Yeah, so blood vessel deformation is, of course, an interesting question, but let's look at that from a physics point of view. You have, let's say in the arteries, you typically see the pressure waves due to the heart pumping, right? And it's actually the pressure that is deforming blood vessels, because if you look at the strength of the material of the blood vessels, that strength is much, much higher than the shear stresses you have in flow, much, much higher. But the pressure is really high compared to the shear stress. So pressure is a normal stress, and the shear stress is a tangential stress. So it's really the pressure and variation of pressure that is driving the deformation of blood vessels. And if you go to the microvasculature, the pressure is much more smoothed out in terms of time sequence. You don't see that much pumping of the blood in the capillaries. And therefore, the effect is much, much smaller than what we would expect on the large scale. I know that some people are interested in that question, and there are some projects looking into this. We in Edinburgh, we have not done that. To be honest, I wouldn't expect a massive influence on the micro scale where you have capillaries. And because a single red blood cell passing by, you have the endothelial cells, they're surrounded by smooth muscle cells and so on. So to what extent can a bunch of red blood cells passing by in a fluid that is flowing, can they really deform the blood vessel? I don't have a full answer, but I would not expect this to be the main driver for what is happening in the microvasculature. Now, I'm not saying that the endothelial cells cannot sense the pressure variations due to the red blood cells moving. That is a different question, right? Mechanotransduction, for example, that's one thing. But deformation, which is a mechanical deformation, I think it's probably not that relevant. That's my personal view.
Zhongzheng says thank you very much. All right. If there are no further questions, comments, maybe let's thank the speaker again and close the session. Thank you everyone online and in person.